Respected speakers, honorable faculty members, dear students, and all the participants joining us in person or online, Assalamu alaikum and very good morning. On the behalf of organizers, I, Dr. Maria Akil Khan, feel privileged to welcome you all in the mini symposium on sustainable agriculture for food and national and nutritional security under climate change scenario. This lecture is organized by Sindh Innovation Research and Education Network Siren in collaboration with International Center for Chemical and Biological Sciences. The main objective of this symposium is to understand how sustainable agriculture plays a crucial role in ensuring food and nutritional security under a changing climate scenario by adopting practices that enhance resilience, productivity, and environmental sustainability. Let us commence our program with the recitation of Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان والأرض وضعها للأنام فيها فاكهة والنخل ذات الأكمام والحب ذو العصف والريحان فبأي آلاء ربكما تكذبان
Subhanallah. So let me take an opportunity to introduce our respected teacher, Professor Dr. Atar Rahman, who is the most decorated scientist of Pakistan and have won 4 civil awards. He had over 1,500 and more international publications in several fields of organic chemistry. And he is the Professor Emeritus of International Center for Chemical and Biological Sciences. Please welcome, sir.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. Professor Reza, Iqbal, Salim Babdi, colleagues, ladies and gentlemen, dear students, Assalamu alaikum. It's a real pleasure to welcome my dear friend Professor Salim. We met a few months ago in Kaust in Saudi Arabia, and I'm glad that meeting has led to this visit, and a strong cooperation has begun between our institution, and I look forward to this cooperation growing in the future. So a very warm welcome to you, dear brother. Well, I was asked to say some words about food security. Over here, the people who are far more knowledgeable than I am, but I was thinking about what we can do together in this institute, in this area. And so I've picked up basically 10 slides, identifying some of the key areas of cooperation. And some of these could also be perhaps, Iqbal, looked at by Comstech to promote so that food security and especially agriculture is a huge challenge with the stress that is now, climatic stress that is now developing with increased global warming the world over. And this is going to be even more of a challenge in decades to come. So we in the Islamic world need to look at what can we do in these areas. So I've selected basically 10 different uh, areas where we could focus on. One of them is the gene editing technologies, which have come in the last 8 to 10 years. Now they're becoming very powerful ways of modifying plant or animal species. So development of stress tolerant plants through suitable identification of genes and then modification of some genes so that one can develop improved varieties. These are projected to have an impact of about $15 billion by the year 2030. So this is one area where we could focus on. Then development of drought-resistant and climate-resilient crops. This is an area where work has already started in this institute. Professor Shahid Mansoor is here. He is leading this effort to look at different modifications, genetic modifications through selective breeding, not necessarily genetic changes, but through selective breeding and development and finding those varieties which are drought and salt tolerant. And this area again is projected to have an impact of over $50 billion within the next 6 or 7 years. The third area is something that we in cities like Karachi and other areas we could do is vertical farming, because in areas where there's limited space and the farming space is limited, you can have stacked layers of farming within these congested places, and you can have year-round production of plants and vegetables. And even the roofs, most of our cities are not utilized. So we had developed a device for producing plants and vegetables just on rooftops, just 15 feet long. And there are 2 or 3 models lying in our institute, which we have tried. And using a small solar-powered pump, we just use a normal light to power the flow of water within a closed circle. So you just have to fill it up once a week, and that again could be magnified. So the roof spaces can be used for vegetable production, and you could have and I think Cropstack again, the top you have a very nice roof garden. So we could put some of these devices there as demonstration units of how one can produce plants. Precision agriculture is now coming in a big way, using artificial intelligence and IoT, so that you have an optimum delivery of water, pesticides, and fertilizers. And these can be, so there's a lot of work going on in new sensor technologies, which tell you in advance how much is it. So we find that in most places around the world, we are using far more fertilizer or pesticides or other materials than are actually required by the plant. And you could cut these to half or 1/3, or in many cases 1/10, without actually affecting the crop production. So, you can boost the efficiency of plant production. This is another area of precision agriculture that we need to produce in the Islamic world. And that again is projected to have a high impact of over $20 billion within the next 3 years of these things. Synthetic biology has come to the fore, and that's again something that could be done very significantly in our own institute with our facilities and infrastructures. And this is all sorts of ramifications in pharmaceuticals, in meat production, for instance, production of artificial meats through various means, and other areas of development can be done through synthetic biology for food production and provision of food security. This will complement traditional agriculture. Microbiome engineering is coming in a big way, and understanding of microbiome and how it functions, and combined with our own metabolome. And so these are areas of growing interest understanding how the beneficial microbes can be used to promote plant health, so and promote sustainable agriculture. So this is again something that can be done in the biofertilizer market. A lot of work has been done in this institute. We've done field trials using neem and other. We worked together and shown that there are many natural plants with activity which can act as fertilizers and also act as biopesticides in particular. So, that can again be an area of considerable potential in the future, especially where there is a sensitivity for developing organic farming. So biofertilizers and use of natural materials rather than synthetics can tackle that. Work has already begun in the institute. In this field, we have projects being now funded by the Ministry of National Food and Security on genomic selection in livestock and crops. And this is, we have already signed an agreement with the Ministry of National Food and Security, who have recently provided us a grant of about 900 million rupees for buying some mass spectrometers. So 6 new mass spectrometers are in the process of being installed in the institute. And this will focus on genetic markers so that understanding the genetic markers through which we can identify desired traits in plants. So this whole, in animals and of course plants, both. It can be used for plants as well as animals. So this is another area of that we could focus on. And then of course, RNAi technologies have come to the fore. And this is a fast-growing field, and that again is something that we can focus on along with biopesticides. Aquaponics is something that we have talked about recently with some ministries, and they're very interested in developing aquaponics here and multi-trophic agriculture. So to have closed cycling where fish production and plants go hand in hand in closed cycles, one providing nutrients to the other. And that is something, again, that based in Karachi, that's something that can be done. So these were some of the suggestions about food security that we could adopt here. I would like to basically this is my last slide, the second last slide. The blockchain technologies for the entire system of production to end usage through blockchain technologies. These need to be brought in to ensure the most efficient delivery of plant materials to the end market. And that's something that we have a lot of wastages because of non-use of some of these modern technologies, and blockchain can serve as a major. So lastly, since we were discussing Professor Salim yesterday about the change of landscape of higher education, I thought I should show you this one slide. This is SCIMAGO ranking of, SCIMAGO is the world's top ranking agency, which ranks countries and also ranks by subjects. And if you look at the bottom here, in the year 2000, this was the ranking of Pakistan internationally in research. 96 in the world in economics, 63 in the world in chemical engineering, 62 in the world in biochemistry and microbiology, 59 in physics, and 48 in agriculture. And the reforms that I told you that I brought in back in the year 2000 to 2008 resulted in a complete change in the landscape of research and higher education in Pakistan. And this is the year-wise ranking of Pakistan. And in 2023, in economics, they've gone up from 96 to now to number 17 in the world. Chemical engineering from 63 to 20 in the world. Biochemistry and microbiology from 62 to 21. Physics from 59 to 23 in the world. and 48 in agriculture, from 48 to number 15 in the world. This is SciMago rankings. And there is no other country in the world which has shown such dramatic changes in the change of landscape. And as a result of this, many pockets of excellence of research have emerged in the country where you have quality research going on because of the number of measures that I took. I think there's something, and this needs to be continued and sustained in the future. We have had some problems in recent years, but we have shown that this can be done. And these are SciMago rankings of the world rankings of Pakistan in research. So if we persist, I'm sure that we can do it. So thank you very much. I thought I'll just give you a 10-minute introduction and background about food security and some of the things that we can do. Thank you very much. I do apologize. I'll have to leave a little early because I have another commitment after Salim Babi's lecture. I'll have to go. My apologies. But thank you again.
Thank you, sir. Now we have Professor Dr. Muhammad Iqbal Chaudhry, Coordinator General, OIC ComSec, Senior Advisor and National Distinguished Professor of Bioorganic and Natural Product Chemistry with us. He's also serving as a Vice President at FOSS and has more than 1350 publications and 94 patents. He is among world leaders that has pioneered contributions in the discovery of novel natural products. So it's my pleasure to invite Sir for his lecture on opportunities for collaboration for food security.
Dr. Salim, QS for coming, Dr. Masood, faculty members, girls and boys. I'm going to give you a glimpse of an institution which was largely established by our teacher, Professor Dr. Dalma. He has served ComSec, which is the Ministerial Standing Committee for Science and Technology for OIC countries for about 16 years, before that. And this is the Ministerial Standing Committee for the entire OIC region. So it is a broad canvas on which we now work, being the fourth Coordinator General of this organization, which was established in 1981, and serving as the second term. This is indeed an honor to strengthen an institution which was established largely through the effort of Professor Dalma. So, given the theme of this particular workshop, I'm going to talk about the opportunities for collaboration in food security under OIC. As you know, Organization of Islamic Cooperation is the second largest political forum after United Nations. It has a great influence and a great leverage in terms of its political dimension and its development in the world. And with this, this is the canvas. I mean, if you travel from Suriname all the way to East Timor, it take you about 19 hours. It's the longest ever flight which you would have. Kindly इसको जरा fix करें. It's the vast land of OIC, where you can see the 57 countries, population of 1.98 billion to be precise, and that is the canvas in which ComSec work. On top of it, we also have in blue color, these are the observer states. Their number is increasing now. Ethiopia has applied the observer state along with Central African Republic and others. And very recently, something very special happened, which is China. China came in with lots of resources and with great intention to connect with a body of 57 countries. It is a politically correct decision that they have in one go 57 countries. Majority of them are part of 4 Belt and Road initiatives. So you're talking about a huge canvas, and there are serious issues of food security. If you come to sub-Saharan Africa, which is called Sahel region, North Africa and West Africa, these are the most food insecure countries in the world. They are arid, agriculture is completely in chaos. On top of it, population is increasing with a huge proportion. And these are among the 33 most most food security vulnerable countries. So this is the canvas in which we need to work along with the organization. This is the only intergovernmental organization which is chaired by Pakistan. So, we didn't really got any UN organization or any other organization. Thanks to our leadership that we got COMSTAC, and chairman is the president of Pakistan, prime minister is the co-chair, general assembly comprised of 57 ministers of OIC, as well as other states, and then of course I serve as the working chairman of COMSTAC. With this canvas of political support, we have the kind of the assembly of this nature, which is now planned for October 25. So COMSTAC has had 13 networks, but among them, these are the most notable one. And if I show you that we have ISNET, which is the Space Science Network in Pakistan, Water Research and Development in Jordan, Oceanography in Turkey, Tropical Medicine in Malaysia, IT in Pakistan, Virtual University Network in Iran, Science and Technology Park in Iran, and Nanotechnology in Iran. Of course, we have others too, but they are the most active. And we also work with cluster of organizations like a UN system. We are the premier organization because we are the pre-ministerial committee. But then along with this, we also have many other institutions which serve OIC community through different domains. And I wish to take you to this Kazakhstan. This is Islamic Organization of Food Security, an extremely potent affiliated institution which worked in the area of food security with lots of funding and lots of networking. We work with them, and we have developed many programs with them along with our own programs, which are both. So this is the mandate of COMSTAC, and we are the implementation body for OIC resolution related to science, technology, higher education, health, and now climate. And we are also the scientific advisory policy. We have revitalized an institution which was established back in 2008, STIO, Science, Technology, Innovation Organization. We have now STIC, which is Science, Technology, Innovation Research Center, and we work at the capacity building. So we really right on the top. Capacity building is extremely important for meaningful in terms of bringing change at the institution level or elsewhere. So we are headquartered in Islamabad, but our functional headquarters is in Jeddah. So we have a lot of interaction with the Secretary, which belong to Chart and has a special interest in food security because Chart Basin, probably all are aware of it, that is the Chart Lake, which is now dried up, one of the largest lakes in the world, is no more there, and the entire area is affected due to that climatic disaster. So this is the COMSTAC portfolio along with our own organization center. We are STIO, which was established by Professor Rahman. ComStech Science, Technology, Innovation Research, Policy Research Center, CSE, which we are working towards getting a charter for a federal university. So this would eventually become a federal university. Act as a think tank providing high quality advisory services for CIA, and also OIC 15 dialogue platform. So this is the canvas which gives us opportunity of working with people within and beyond OIC. We are also custodian of OIC Summit. See, OIC Summit takes place on political matters, and OIC Summit only on the subject of science and technology is being organized. And OIC ComStech is responsible of preparing the agenda for this particular meeting. And the next meeting is going to be planned in Baku, Azerbaijan. So we are working towards that. We also chairing the steering committee for CSE 2026. We have, like UN, we also have our own sustainable development goals, which is called Action for 2026. And these are various ministers which work under the steering committee, and we have been organizing this meeting twice a year. So these are the priority areas in which we work, and just take you to relevant part for security and expectation of OIC water provision. We are the custodian of OIC water provision, safety of water, food, and environment. We work with water provision, managing energy requirements, environment climate change. And then a very important thing happened in the last 5 years, that we have decided to use the collective strength of the most scientifically advanced OIC nations. So you can see that these are the countries selected by transparency criteria, and by virtue of their scientific advancement, they are part of a group which is called OIC. And this is very important because these are the ComStech focus thematic areas. There we work with various organizations and focus on certain elements of development endeavors which are directly relevant to environment and food security. So these are some of the key initiatives of ComStech. Because I should remind you that we do not only work at the national level, it is just part of a very small domain, but we work with at least 32 countries in this particular program. So this is the landscape of food insecurity in the Muslim countries. Around 733 million people face hunger in 2023. That includes Pakistan. Pakistan is among the 10 most food insecure countries of the world, and the 4th one among the list. So imagine a country with the size of our world with tremendous resources. That is a gross agriculture failure of research and extension. And there we need to work. So around 233 billion people globally face moderate to severe food insecurity, among them largely Muslim countries. 2022, around 203 million people were undernourished, largely in Africa. About 100 million population in the Sahel region, which is between the tropical and Saharan Africa, is where we have food insecurity. So what we have done was that we have established the only OIC organization, which is called ComStech Forum on Environment and Ecosystem Restoration. It is UNEP, United Nations Environmental Program of OIC, and under this, it was launched in 2022. We had many activities. So, for instance, we had very last seminar on land degradation, and a report was published, submitted. International Virtual Seminar on Solution to That, Food Insecurity and Malnutrition in Sahel region. International training workshop on addressing challenges of food security in Sahel at Niamey, Niger. International training workshop on related practices for development of sustainable businesses in Benin, West Africa. Along with this, we also have 25 large international organizations, including UN organizations, part of the network. And this provides us the opportunity of conducting many important events. So in September, we have a food system transportation at Makerere University, Uganda, which is the worst country. International training workshop on resilience, smart cities at Elanga University in Indonesia. International workshop on food security challenges in Mogadishu, Somalia, and international training workshop on the system registration, Jakarta. And you are all invited to be part of it. We have 100 slots, which is called Constant Youth Leadership Program in these areas. And that we have many youth leaders who get all support to come and work in complex area. Right now, we have residents of 10 countries working in complex area. And they work on leadership skills. You know, how to lead international program, because this is the multilateral organization, which is here to give you training on advanced leadership skills. So, scholarship, fellowship, and training. I'll tell you that ComSec has the largest scholarship program, much larger than UNESCO. So we have 5,000 fellowship Palestinians, 700 fellowship for refugee scientists, 150 fellowship for men-in-reach scientists, and many more. And among them, this is also a fellowship scheme especially focused on nutrition, food and agriculture sciences. We also have ComSec Industrial Leadership Program. With Gourmet, we are conducting a large internship, and every year we receive about 20 of these fellows to work in gourmet industry. This is a very important forum which we have initiated about 6 years ago. It's called the Youth Forum on Agriculture Biology. So the 4 of them will be held in Dhaka, and that is specially focused on agri-biology. So it is largely young people coming up with the ideas of agriculture. Then we have halal product testing and standard programs. So we have been working with many countries of the world of developing their capacity in the field of halal food production and testing. And with iccbs, we launched a major program of about 1.5 million USD. We're spending about 300 million annually in 26 common countries of iccb. I'll tell you that iccb is one of the institutions which was established by late Professor Abdul Salam. It's the International Center for Genetic Biology and Agriculture. So we have been giving research grants, research fellowships, and courses. And of course, this is a lot of documentation which comes from ComSec. ComSec Award are legacy awards established back in 1994. They are awarded by the President Pakistan. We have OIC Technology Portal. So we are custodian of technological development. And if you visit this, you will find a lot of technologies relevant to security. We're also working on network water centers. We have recently established a halal product center in Uganda. there will be a lot of documentation. So I'd like to thank you and wish to tell you that opportunities exist, and I think these are the areas in which we can have huge impact in OSCE. So the ideas which are given by our teacher, Dr. Karama, will be implemented. You are listening to that. Thank you, sir.
Thank you, sir. Now we have our respected foreign speaker, Professor Dr. Salim Al-Baghdadi, who is a renowned researcher at King Abdullah University of Science and Technology. He has several publications to his credit, and his work focuses on plant biotechnology, particularly in enhancing crop nutritional value and stress tolerance. Please join me to welcome him for his lecture on the topic of From Bench to Field, Translating Research on Plant Metabolism into Agricultural Solutions.
It's a great honor for me to be here and to have the opportunity to present part of our work that we have been doing at Laos in the last 10 years. I want, of course, to thank Professor Ataur Rahman for his visit to KAUST and for paving the way to this collaboration that we are currently having with Professor Shahid and Professor Farouk. I hope we will continue and expand the collaboration here and build a quite stable bridge between your institution and KAUST. You see here, KAUST is a small university, is a young university, has been established in 2009. It is rather a research institute than a university. In my talk, I will have a brief introduction, then I will explain our work on combating striga, that is a root parasitic plant, devastating cereal production in Africa. Then I will present shortly the discovery of a novel growth regulator and how we established mimics of this growth regulator for agricultural application. What you see here are beautiful colors. They are the result of accumulation of a certain pigment that's called carotenoids. Carotenoids are widely spread in nature. We know them from these colors, but their major function is actually photosynthesis. They are protecting from photooxidation and contributing to the light harvesting process. You see here the pathway. I'm not going to go through it, but important to know is that green tissue is accumulating different carotenoids. You see here the structure of some of them. We have special tissue that accumulate particular carotenoids, such daffodil flowers or tomato that accumulates lycopene. Now, okay, that's fine. The conjugated double bond system that's responsible for the color of carotenoids is also responsible for another feature of them, which is susceptibility to oxidation. So every double bond in this conjugated double bond system can be cleaved by oxygen. Result are cleavage product that we generally call apocarotenoids. The cleavage product can be triggered either by reactive oxygen species or by specific enzyme that plants have evolved to cleave particular double bonds in this conjugated double bond system. Now, the cleavage products are not a trash. They are not a biological trash. They are important metabolites. Retinoids are some of them. You know, we have retinal in our eyes for vision. We have hormones, growth regulators, and we have, volatiles, pigments, and so on. I started my career working on metabolic engineering to increase pro-vitamin A content in rice. You know, vitamin A deficiency is still one of the major nutritional problems in the world. It's particularly present and prevailing in countries where rice is a major staple food. Later on, I worked on elucidating pathways of hormones. One of them is strigolactone. Strigolactone regulates branching. That is the major function. If the plant is deficient in strigolactone, it becomes dwarf and it develops more tillers or more branches. Strigolactones have, of course, other functions in regulating growth and development in different tissues like other plant hormones. They have another function. They are released in the soil. In the soil, they attract partners for symbiosis, mycorrhizal fungi. This mycorrhizal fungi would colonize the plant roots. Their hyphae that are spread in the soil would absorb minerals and provide them to the plants. The fungal partner is rewarded by photosynthetic products that are coming from the plants. The plant feeds, in principle, the mycorrhiza to get the minerals and water from the soil. Now, strigolactones were originally discovered as a rhizospheric signal. This means a compound is released by plant roots and is able to induce the germination of root parasitic plants. Seeds of these plants remain in the soil dormant. When they perceive the signal coming from the plant, they would germinate, build a structure that's called haustorium that builds a bridge to plant roots, allowing the parasite to siphon waters and nutrients. The result is a host that is suffering and a parasite that is blooming. Now looking at the structure, you see here the different types of strigolactones. I'm not going into details here. Important to know is that we have canonical and non-canonical strigolactones, and that is an analog that has been used in the biology since many years to elucidate strigolactone biology. Now, when we started working on the pathway, there were 1, 2, 3, 4 known enzymes. Paper chemistry told us that to make strigolactone or carotenoids, you need around 9 steps to make this moiety here. And people thought that the other moiety, the D-ring, is coming from a different origin. The final step is coupling of the two. And we know, as I said, 4 enzymes to carotene cleavage dehydrogenase, this one enzyme of unknown function, and a cytochrome that is in the cytosol. We elucidated the pathway and we found that nature is much more clever than paper chemistry. In principle, we need 1, 2, 3 enzymes to get to a structure that we discovered and we called carlactone. The merit of the short pathway is actually coming from this CCD8 enzyme that can catalyze different types of reactions simultaneously, is also quite stereospecific. Carlactone is, in principle, the precursor of all strigolactones in nature. That is the strigolactone biosynthesis as we know today, quite complex. You see here the central role of carlactone and its derivative carlactonic acid. A plant synthesizes and releases many types of strigolactone. Now back to striga. Striga is a root parasitic plant and is considered as one of the major global traits for food security. It's particularly important for African countries where it's affecting 32 African countries. We have around 50 million hectares infested by these root parasitic plants. Economic losses go from $7 to $12 billion annually, and it's severely impacting the food security of 300 million people. You see here a short movie about striga. You see the huge devastation that's coming from this root parasite plants. The parasite is blooming, flowering, and the host, the cereal in this case, is suffering. Now, one major constraint if you want to combat striga are the seeds. because a single striga plant can produce up to 200,000 tiny seeds that remain viable in the soil for more than a decade. You see the size of the seeds even after magnification in comparison to the seeds of the host, of the cereal host. Actually, these seeds are spores rather than seeds. Now, one theoretical option to deal with the striga problem is the so-called suicidal germination. As I said at the beginning, striga needs a signal that's coming from the host to germinate. If there is no signal, it would not germinate. So we thought, why not using this dependency to develop a strategy in which we apply strigolactone analogs in the absence of a host. These analogs would induce the germination of the striga seeds, but the seeds would die because these are obligate parasites. If there is no host around, they cannot survive. Now, to test, establish, and assess this strategy, you need several things. The first one is you need efficient and affordable strigolactone analogs. You need suitable formulation, and you need an application protocol for rain-fed agriculture, which is the major type of agriculture, particularly in the Sahel zone, one of the poorest regions in the world, where water is also scarce. Now, when we investigated and discovered calactone, we found that calactone itself is actually active as a strigolactone. You see here its ability to induce seed germination in striga in comparison to the classical analog that has been used since decades. It is inducing seed germination, but it's less efficient than GA24, the strigolactone analog. But if you look at the structure, you recognize the simplicity of calactone compared to GA24. That is a big advantage that we wanted to build on. So we developed based on calactone, strigolactone analogs. You see here the structure of the first one. This was synthesized by a Swiss company, which we call nitrophenlactone. We tested it. We got activity, but not a high one. Against Striga harmonica, the activity against another root parasitic plants was higher. It was not the final product, but it told us that we could continue using this strategy. We were lucky. Mr. Gates visited CHAOS. We discussed the idea with him, and he agreed to to support us in establishing this strategy. So we started around November 12, 2015 in Burkina Faso. Developing strigolactone analogs, assessing them, and testing them in field. In this project, we were also looking at resistant at genetic level. For this purpose, we used permalate as a host, and here you see two contrasting lines. The one is affected by striga, the other one is resistant. We looked at the two contrasting lines. We provided very high platinum level genome, and this actually led us to the identification of the gene that is responsible for the susceptibility of the commonly used variety. Now back to chemistry. So we developed in collaboration with Professor Tadao Asami at the University of Tokyo different strigolactone analogs. You see here the first generation. We established an activity workflow. We tested first the activity in the lab. See here, when we applied the analogs, we got seed germination. We went one step further to the greenhouse, tested the activity. Then we established that what we call mini box, and then we did through filters. For the high throughput assessment of the strigolactone analogs, we use AI tools. We established a sequent deep learning method that allowed us to screen very quickly the activity of the strigolactone analogs. We also established a protocol for rain-fed agriculture. Idea is we wait for the rain, we apply after the first rain our strigolactone analogs. This rain, the second rain would then enable the distribution of the strigolactone analog in the soil. Other rain will continue distributing the adjunct in the soil. Result is then inducing seed germination and death of the seeds. You see here one of our visits to the field there. Dr. Jamil has been actually a major central player in all the stories. He makes at least 3 visits to Africa per year. You see here results of the first field trials. They were very promising. You see here 3 strigolactone analogs, reduction of infestation by 65% in vermouth, by 55% in sorghum. We selected 2 analogs to continue with based on their activity and their simple synthesis. We got them in collaboration with a company, synthesized at a higher scale and also formulated. We tested them in Kenya now. You see here the results of the trials. We got another formulation in granular form. Here again, you see the results, reduction in the striga infestation. We assessed, of course, the safety of the compounds. You see here the results of the AMIS test. That is actually a simple test, and it's a first test that you would do if you want to assess the safety of a compound. The results show that the 2 compounds, the 2 strigolactone analogs that we are working with are not carcinogene. We assess the impact on the soil microbial community. Here we saw a change that is not stable, but that change is actually in favor of the plants. We increased the number of cyanobacteria in the soil. And we got last year fund for other 5 years. In this 5 years, we will optimize the production cost, optimize the application protocol, enhance the efficiency of the strigolactone analogs by mixing them with a different type of seed germination inducers. And we need, of course, to do feasibility, efficacy, and safety studies and go to registration. Now I'm coming to the second part of my talk. It is about Zaxinone. The story started with the discovery of an enzyme in rice that makes this reaction, giving rise to this molecule that we call Zaxinone. It is, here you see phylogenetic tree of CCDs in plants. This enzyme represents an overlooked clade of these enzymes. The family is providing, as I said at the beginning, phytohormones, abscisic acid, and strigolactones. We believe that Zaxinone is a candidate for a precursor of novel plant hormone, or at least an important growth regulator. Now, if a plant doesn't have, a rice plant doesn't have the Zaxinone synthase activity, this, you see here the mutant, you see the effect on the growth of the plant compared to the wild type. We confirmed the biosynthetic map that we obtained through in vitro assays by showing that Zaxinone, the product that we have, is actually present in plants. You see here comparison LCMS identification. We quantified the compound. We saw that the mutant has less amount than the wild type in the roots, which might be the reason for the observed phenotype. We confirmed that this is the reason by exogenous application of Zaxinone when we apply it to plants that are grown hydroponically in water. You see that we can at least partially rescue the roots phenotype, the growth retardation. We were also surprised to see that it is growth promoting also in wild type. That is the results in hydroponic, where you use quite hormonal concentrations of 2.5 micromole. In the soil, we increase the concentration of 10 to 10 micromole. But here again, we saw the growth promoting effect of Zaxinone in wild type and its ability to rescue the phenotype that we observed in the mutant. Now, we did also a small field study. There we noticed that the mutant is actually has a tillary phenotype. It has stillers. This indicated that we have probably an increase in strigolactose. As I said at the beginning, this hormone is regulating filaring. Indeed, we saw that the mutant has, see here, the zaxinone mutant has higher level of strigolactose, which explains why it has less telomeres. I'm not going into details here, just to let you know, zaxinone is regulating the biosynthesis of strigolactose at transcript level. We did several studies at metabolomics and transcriptomics level, and also we looked at the events that take place at cellular level in the roots. You see here a summary of the mode of action of zaxinone. Our data indicate that zaxinone is improving photosynthesis, increasing sugar transport to the roots. In the roots, you have a higher conversion of energy that gives that what is required for better growth. At the same time, we are changing the cytokinin level, we are changing the strigolactone. Now, what happens if we overexpress the gene? You see here the phenotype that we got upon overexpression is a clearly increased yield. That is a result of increased number of productive telomeres. Again, I'm not going into details here. We got, in principle, an explanation for the high tellurine phenotype. Again, back to strigolactones. We looked at the roots. We saw increased meristem size, which explains also the better growth. Metabolomics analysis again showed us that we are increasing sugar metabolism, and we are improving photosynthesis. Very important, we got increase in the uptake of minerals. You see here again a summary of the effect of the gene, now not the compound, better uptake of the nutrients, better photosynthesis, increased sugar metabolism, and the result is more telomeres, and these telomeres are productive. Now, if you want to apply zaxinone to fill, that's the natural compound synthesis is a bit complicated. So we thought about developing mimics of zaxinone. At the beginning, we did that what we call structure-activity relationship study to know which components of the molecule are important for the activity. We know that the all-trans configuration is important. The length of the chain is important. The hydroxy group is less important. Based on that, we thought about developing mimics. The idea was simple. Many people are replacing this isoprene unit by a phenyl ring. This was shown to work in developing of insecticides in principle. So we use this principle and replace the isoprene unit by a phenyl ring. Tadao Asami got the idea also of inserting an oxygen in between them. We got 5. That is only the first generation. We generated many other mimics of them. We tested them and we found that MESAC3 and MESAC5 are actually quite active. That is, you see here the effect on transcripts of strigolactone biosynthesis in comparison to zaxinone, the effect on strigolactone quantification. You see here the effect on the growth of roots in soil. That is zaxinone, and that is MESAC3 and MESAC5. So the two compounds are at least as effective as zaxinone itself. We tested the two compounds on different crops in different trials. I'm just showing here a few of them. That is an experiment that we did with pepper. You see the increase of yield when we apply the MESACs. In comparison to humic acid, our compounds are much better. We also did nano-encapsulation of zaxinone and repeated the pepper experiment, got again increase in yield. And importantly, we got an increase in the zinc content because the nanomaterial that we use is the metal, is MOF, that is metal framework, which contains zinc. As I said, the compound increases the uptake of nutrients, and zinc is one of them. We did a field trial in Taiwan with paddy rice and also upland rice. You see here with Mizax 5, you see the increase in tiller number, panicle number, panicle fresh weight. And in this experiment, we used 50% fertilization. This means that is the evidence, of course, for the activity of Mizax 3 and Mizax 5 in reducing the need of fertilizers. We know the huge impact, ecological impact of fertilizer. That is another field trials, although on paddy rice here we compared 100% fertilization with 50% fertilization. You see again here the effect on tiller number and shoot fresh length. So on the manuscript has been just actually submitted to Nature Sustainability. We didn't see an effect on photosynthesis in the field, in contrast to the data and rice. Very important, we did not detect the two compounds in the rice grain. This means the rice grains don't have them at all. They are not transported up, which is quite important. I included these slides just to show the priming aspect. We were applying Mizax either by spraying or by irrigation. That is another mode of application, which is of course much easier. Putting the seeds in the solution that contains Mizax, prime them, and then plant them. We did that with rice, priming for 24 hours. We put the plants in the greenhouse, and you see here the effect on the height, number of tillers. We didn't complete this study up to yield quantification and phenotyping. That is seed priming with tomato. Again, you see here the effect on shoot length, on root length, when we just put the seeds in the solution and plant them. With this, I'm coming to the end of my talk. I have to thank my collaborators. And as I said at the beginning, I'm really very glad and very happy that we will build here a strong collaboration with your institute. I have, of course, to thank my team. As I said, Jamil played a major role in the spider story. That is a short movie about our work. Thank you very much.
Thank you, Professor Salim, for such an interesting lecture. Now, I would like to invite Thank you, Professor Salim, for such an interesting and insightful lecture. Now I would like to request Professor Dr. Shahid Mansoor, HEC Distinguished Professor, Senior Advisor, iccbs, Ex-Director, NICG, and Fellow of Pakistan Academy of Sciences, for his lecture entitled The Concept of Regenerative Agriculture and the Role of iccbs team in Enhancing Conservation Crops. ये बंद कर दीजिए।
Okay. Okay, first of all, Bismillah ar-Rahman ar-Rahim, I must thank all the distinguished guests that have, attending this mini symposium. We invited Professor Salim Al-Babli and Dr. Jamil, and we thought that, we should also discuss what we have been doing in the last few months in Pakistan. And also, I must thank Mashoor Saab, Asif Saab, and also some of the farmers and students who have done a lot of field trials. And, I must say that they have actually done a lot of work and a lot of appreciation for all this hard work. And especially to some of my colleagues, Walid, I must acknowledge that they have done a lot of very good work to actually so that we can have some data. So the concept that we are trying to do promote here is regenerative agriculture and how we can help in all these activities. So let me come to the one point that all over the world, including Saudi Arabia and Pakistan, now we have been asked, researchers have been asked that, what is the impact of research that we are doing? We are doing wonderful research, but, is it coming into practices, what we call as innovation and commercialization? So innovation, Professor Rahman has said that, we have done a wonderful work in enhancing the publication in this country, but, still we are lacking in innovation index. So I think that, this is something that we need to think about, how we can enhance our innovation index. Coming back to the agriculture is in stress in Pakistan for many reasons. One of them is that, is population, water, climate change, land, pesticides, and disease and salinity and waterlogging. And one thing which is added to all this scenario is the cost of production. And the yield and the price that farmers are getting at the moment is very, very dismal, which means that farmers are really struggling. Yesterday we visited a farm, and then there was no water for growing cotton, so it was near Gharo. So I think that we have a lot of challenges. So we need innovative solutions to come up with what are the possible solutions that we can have. So obviously, one thing that I think that my success there in the last 2.5 years is that I've been able to convince iccbs that food security is an important issue. So, and because we were mostly focusing on health issues, but the largest disease of this country is food insecurity. Hunger is the largest killer. So if you have, and also it is adding up with the nutritional problems, because although we are sometimes we are able to feed enough food, but it's not nutritional is a question. We are deficient in minerals, vitamins, and protein. And that's why in sustainable development goals, the top target is food security. So I'm glad that people sitting in the front row spoke about food security, which is very encouraging. So we have all observed climate change. We have floods in other parts of the country, and still the Bela Dam is still not filled. That's a tragedy. And then we have farmers waiting in Sindh. We visited one farmer that he is still waiting for water to transplant his rice nursery. So we are struggling. So this is what we call as climate change, and this is affecting lives all over the world and very much in Pakistan. One victim of this climate change is cotton. Why we want to grow cotton? Because it's a crop that has fiber, it has food in the form of oil, and it's a feed because the seed cake, and then the leftover is used as a to support cooking. So this is a whole scenario which has changed when we have reduction in cotton. So this was, I mean, this is going down, and now last year we had only production of 5 million bales, and this year we expect that it may go further down because of the late sowing of cotton in Sindh province. So there are obviously solutions which are beyond our domain, like water reservoir, rainwater harvesting, cropping pattern changes. So this is something that as a scientist we can do. We need to reduce the cycle of crops from 6-7 months to about 4 months, because when you have shorter cycle of production, that means that you can use resources much more efficiently. And the most important thing is genetic gain. Obviously, we work on genetics to improve plants and livestock for higher productivity. So thanks to some of my friends sitting here, we have the concept of regenerative agriculture, which means that that we are now understanding that we have done a very bad practices over the last 40-50 years, is that, and this has happened all over the world, that we have destroyed the ecosystem. The soil health is badly damaged, and we understand recently that the soil is a living thing because it has a microbiome, and so regenerative agriculture is the future, because we want to reduce the input of water, pesticide, fertilizers, and this is what it should result in: enhanced water use efficiency, control of plant growth through nutrient growth management, and better genetics, biofertilizer, biopesticide, and biostimulant. So I think that I will give a little bit about details about what biostimulants we have recently tried in our company. So about 2.5 years ago, we started to work on purification of non-GMO varieties, and I'm very glad that it was a very small project which was funded by Artistic Millionaire. The results have already gone into the field. So we have distributed pure non-GMO seed to at least 3 seed companies. So that task is accomplished. Now they are multiplying. I'm just trying to help them so that they maintain purity. Root phenotyping is a very important aspect, as Professor Salim has also mentioned, that root is a very important component of the whole plant, and some plants have deep root, like cotton. So if the plant has a good root system, that means that it is much more able to cope with the stresses. And then we optimize conditions for multiple generation, and then we establish the system that because of the mild weather in Karachi, we could grow 3 generations of cotton in 1 year. This was very important because when you are doing seed purification or generation advancement, you can reduce the time required from 4 to 5 years to about 2.5 years. This was the MOU that we signed, and then we have this major success in developing cotton or having 3 generations in a year. By the way, it was all done in natural conditions. One thing that we have here at iccbs is a genome center where I'm placed. So the new requirement by the for intellectual property is that you need to have whole genome sequencing of the varieties that you are claiming. So we were able to identify markers for the varieties because we did some purification selections, and by using SMPs, we were able to identify some markers, and the other requirement of the industry was to have uh, to have uh, uh, to have a single stem variety which can be used for machine picking. So this is something which was required by the industry, which was funding this research, and we selected some plant, and we are now this seed is being multiplied at various locations in the country. Now this is something which is very important, is that there's a huge problem of one insect. As Professor Salim discussed about striga, we have a problem of pink bollworm. It's a big devastating disease, insect, and when you go into the field, you have all these pink bollworm. So we were able to generate double gene and triple gene cotton. I patented this cotton, and we did expression studies and then field trials. And this is a very interesting site, and it was taken yesterday. So normally the ball weight is about 14 or 15 gram. Here what we are getting is about 26, 25 gram. So it's just like a अमरूद की जितने बड़ा जो है ना ये ball है. And this is in Uttar, so you can visit and observe yourself. So this is, I mean, I was very surprised that, when we went to Uthal and did this in the real field condition where, so I think that the other thing about, as I discussed earlier, was about the staple length, which was the standard for Pakistan is 28 millimeter. We are getting 32 millimeter, and also the genetic outturn is also very good. So I just summarize what Professor Salim has just discussed, that there are two biostimulants, and we so very kindly he supplied us with two biostimulants. The question was that how we do this research. Do we want to just do a small pot experiment, or do we want to have a field trial? And because of the limited funding, and so instead of involving government agencies, I actually involved some of our ex-students who are also farmers. So 3 of them are sitting here. 2 are from Bluestan, very proud of them, and then 1 from Mushairofiro. And then we have all other students who are very, very keen to work on this, and a lot of students came from the agriculture department. Thank you very much. And I would say, so now I'm actually overwhelmed by the student from agriculture department. So everybody is asking that how many students you have. But so the experiment that we did, and mostly it was done by Waleed, is to have dose optimization, Waleed and Hamza. And we did dose optimization by 2 ways. So we can have 2 ways of applying them. One is by irrigation, which means that we give them with along with water, or the other one is by spraying. So we tried both, and this experiment is almost done because we started earlier. And then field trials at multiple locations. So because of the links that I have in the country, some of them are sitting here. We were able to have trials in Lahore, Faisalabad, Multan, Bihari, Bahawalpur, Vania plant in Punjab, Sindh, Karachi University. We have a small trial here. Surti Artistic Fabrics, Nushairo Firoz, Mirwa. And we are not paying to, most of them are not paid. I mean, they are just courtesy there. Bulasan, we are doing Uthal, Bela, Panjgur, Turbat. At Panjgur and Turbat, I'm paying these guys because I thought that I need to have a very good data, so we are just paying a little bit of money to them. So this is the result so far. What we see is that as compared to control, we see increase in the plant height, and also you will see that so the control is in green, and then you have and these are the name of different varieties. We use actually some of the varieties from a commercial company. These are all GMO varieties, and the largest trial is done at Uthal with Ravi Saab, where we are using 13 varieties. The other very interesting impact is in the number of bowls. We are seeing, but because this is ongoing, so obviously we still have around 2 months to go, and so we are seeing increase in height as well as in number of bowls. So what I suggested to Danish and Kambar to have a gathering of farmers at their field and show them that how this small treatment can help farmers to have more yield. Now, Professor Atavarman just mentioned that we have new technologies that are available, and one of them is what we call as CRISPR. So CRISPR has emerged as the most important technology for modifying the genome, and luckily, I will share you one thing that, but luckily we have a minister, federal minister on climate change, and he just chaired up and formed a committee, and I'm also a member of the committee. So I think that we are going to have a commercialization of this genome edited technology. So we submitted a policy framework for commercialization of this. But it's very important that that we develop as a scientist technologies, and some approvals are required by the government, and a positive attitude towards science, because that is something which is very important for having an impact in the field. So one thing which I was very surprised that whole livestock industry was very interested in low toxic gossipol cotton. I just got some lines from China through my friends, and we analyzed them, and then the one thing that we were very fair, we have a fear that it may be susceptible to a lot of insects, but there was no difference in pest infestation. We are trying to publish this. And also with some of the collaborators, we were able to do some toxicology, and there was no impact of. So if you overdose these toxic gossipol containing cotton seed cake to animals, it has some impact on fertility. But when we are using this toxic gossipol less cotton, there was no impact on fertility. So I think that this is very safe, and we should go for. So what we are doing is a process of toxic gossipol edited cotton into our local varieties, and I think that the feed industry is very much looking forward to it. Now, this is something that we have published very recently. So there are four negative regulators of yield. So yield is controlled by four traits: number of tillers, so that how many branches that you have. I hope that you understand that what are tillers. Number of grains. So if you have more number of grain, you are going to have more yield. Third one is grain size. So in case of rice, we actually want long grain, not the thick grain. Otherwise, it will ruin the acceptability of the grain, because we don't want thick rice, we want long rice. And then we have one more negative regulator of grain side. Although in case of wheat, we actually want to have a thick, healthy, mota sa dana chahiye hota hai. So we were able to get this. So we had so most of the important change that we saw when we did this in the glass house was the number of tailors, and the number of seed is actually you can see as compared to control, which is in the bottom. What is happening is that you have increase in the length of grain, which is very interesting. It could have been other way around, but luckily we were. So this has been published, and we in basmati rice, very amazing result in the field condition. We got 124% increase in basmati rice. Basmati is a prime rice, but it produces less than normal, what we call as mota chawal, जिसे हम कहते हैं. उसके मुकाबले में, because it produces more grain. So I mean, the price is almost double than the non-basmati rice. And we hope that with the ministry and ministers are very much interested in commercialization. Another very positive thing is that we always compared ourselves with India. So India has already commercialized 2 genome-edited varieties, and we hope to have a very positive outcome for Pakistan. The next is wheat. Wheat is our staple crop, and again, we have done some genome editing in wheat, where the number of tillers has increased. And obviously, when we have higher tillers, you have higher yield. We have done some trials recently in Karachi, and the results are amazing. So we are just trying to make sure that the final wheat is free of any transgene. This is another experiment which was done. So these are BNI. BNI is biological nitrification inhibition. This is a very important trait for nitrogen use efficiency. So if you have BNI lines, you are very likely to have very efficient use of nitrogen. What we have done here is in the field condition, and the field at Karachi University are where we are growing wheat is highly saline. So this was by chance that we were growing this with the full synthetic, half synthetic, and symbiotic. This is a combination of different biofertilizers. I've not seen such an impact of biofertilizer on the yield. I think this because this biofertilizer, which is a combination of different microbes which fix nitrogen, or they are also helping plant to overcome stress. The root growth is better, and what we see is a very good yield increase when we so no chemical fertilizer at all. And this was in a field which was not cultivated before, so I think that the results are quite reliable. Very good for organic cotton and wheat rotation. So I think this is something which should be commercialized in Balochistan as soon as possible. This was done in pot, again the same data. Another thing that we are trying to do is GI, geographical indication based branding of cotton. So India has some brands of cotton like Kasuri cotton. So what we have done is that we have done, while we were doing the field trials for our biostimulant, we were very cleverly integrated this into a field trial for varieties. So we have now 13 varieties growing in different parts of the country. And what we are doing is that we are also uh, hoping that, by the end of the day, we'll have the data on the quality of cotton, which means that, fiber length, fiber quality, strength, and, fineness, all those data. And again, our friends are helping us to do fiber analysis. So very thankful to them. So we have uh, so some people are offended by that, why we call this as a Balochi cotton, because it's coming from Balochistan, so there's no harm in calling this as a Balochi cotton. It's actually a term already used. So we can have other cottons like, Cholistan cotton or Thar cotton, because, it comes with a GI tag, the graphical indication that when you grow cotton or any product, like this is like basmati rice, there is a region in Punjab where when we grow rice, you have that quality. And so this geographical indication will be a good idea to have high return for farmers. This is another very interesting project. Where we are trying to develop, again, this demand came from the industry. I mean, they were using very crude way of developing plant extracts for this biopesticide. And there was no standardization, no formulation. So, they were just boiling or just, making an extract. I went to Rakni at that time I took picture, and I thought that, we can do something about this. So luckily we got this project, and we are extracting three things: Citrullus polycins, Calotropis, Rhiza stricta, and later on I developed a very good collaboration with Professor Dina Siddiqui. I thought that, she would have wonderful collaboration, and we are using her, what we call as a pilot plant to produce some of the extracts. It would have not been possible to have the extract with the level that we are producing now. So we can have a huge quantity of extract made from these plants thanks to her. And we are using neem as in because we have to compare apple with apple, not with oranges. So the same way, we are using biopesticide, which is neem based, is already listed with the plant protection department. So what we are doing is that, we are using that as a standard and comparing them with the. But there's a lot of huge molecular biology and metabolomic work which is going on. So we have Dr. Atia here. So what we are trying to do is actually to understand the metabolic pathways that are involved in the synthesis of these very important compounds which are produced by the nature. And also, obviously, with having Dr. Dilshad, we are trying to have different formulations so that, it is stable for the, at least for the life of the cotton crop. And so these are some of the very wonderful work that you can do, while you are doing applied work. So to Dr. Atia, I said that, you should have several nature and science papers on this. So this is the way that, you progress science. And the idea of biostimulant came also from the basic research. This is another very interesting possibility of using an insecticidal protein. This is a very small protein, about 29 amino acid long. And we are expressing this protein, and we have done some previous work. Now we are trying to express this protein in yeast. The idea is that, either we can use yeast directly to spray on plant, or we can make extract and then do formulations and use this as a. So this has a lot of potential to be used as a biosafe biopesticide, because we are not engineering plant. We are just using an insecticidal protein, which is very well documented. As we have done a lot of work on the biosafety and toxicology of this protein. So there's no effect on the other useful insects or human. So I think this is something which is coming up very nicely. Uh, so there are, just like, we are getting old, so we tend to give a lot of advices. So I thought that, we can give some advices. We need to work on innovation index. So our previous generation has worked a lot to enhance our publications. So now we need to work on the innovation index. So we have technologies available: genomics, genome editing, genomic selection. I showed you a few examples how we have used genetics to actually, have data which is required. Cotton variety suitable for organic, regenerative, and conventional cotton. I think that all our cotton should be regenerative. But I think that this is coming up very nicely. But this is something which is more kind of possible, where we try to use pesticide, try to use fertilizer, and try to use water. And by the way, if we do all these things, we can have carbon credits. IW32, this was a demand from the industry. This is Dr. Shakir is not here, but we are close to achieving this. We are proposing cotton branding with better varieties and geographical indication, because that will help farmers. And use of biostimulant, biofertilizer, and biopesticide, improve yield and nutritional value in crops. Very, very important. Dr. Salim just discussed about zinc. I think that we should think about the iron as well. If we can, so one of my students has done some work where we have used nanoparticle to enhance iron in the grain, rice grain. I think that this is very important for a country like Pakistan. I forgot to mention that we also have engineered tomato for higher vitamin D, and we have one of our students, Daima, is trying to enhance both vitamin D as well as the yield of tomato, because we have to buy seed all the time. But so if you can fix the hybrid vigor, and then we don't have to buy a seed every year from these companies. Also, we have to have climate smart agriculture. I think it's and also better international linkage. So thanks to Dr. Salim, who is here, and also greater link with government and industry. That's very important. You need to talk to. One day somebody will listen to you. I just give you two examples. One was a 900 million investment as a federal Ministry of Food Security at the institute, and then the recent development of support for biotechnology products by the Ministry of Climate Change. So I think that we need to talk to them and industries. Industry don't have time, so if you have lectures for whole day, they will be bored and they will say, okay, we don't understand, and they want something that helps them and enhance quality for traceability. This is something which is ongoing, and we need some more investment in this area. So this is the demand of the industry, that they want to know how the cotton was produced and where it was produced, and then we need to have metabolic markers so that we can have traceability of how the product was made. And I think this is I already tried to not to forget names, but obviously there are a lot of people that needs to make knowledge, especially Professor Atar Rahman, Professor Raza Shah has just entered, and then King Abdullah University of Science and Technology are providing us a very small grant as well. Not very small, but it's a moderate grant. I hope that funding agencies are always, like TDF, ICCBS leadership, that we have understood the message that I've been trying to convey, that there are certain areas where we can have convergence of good chemistry with biotechnology and genomics and then that can create an effect on society. Thank you very much. So, so question answer at the end of, I think we have one more talk.
Thank you, sir. Now I would like to invite Dr. Farooq Ahmed Khan, who is an assistant professor at Third World Center for Science and Technology at iccbs. His work is mainly focused on organic synthesis, particularly in the field of advanced synthesis, organometallics, and nanocatalysis. Let me welcome him for his lecture on the topic of control of lepidopteran insects by using mating disruption technology.
Thank you very much, Dr. Mavia, presentation. So today I am going to talk about pheromones. So how many of you know about the pheromone? तो खुशबू के बारे में बात करने जा रहे हैं हम, लेकिन ये खुशबू इंसेक्ट से है और so this can be controlled by insects with the help of pheromones. So there are some insects which are a major problem in agriculture, like moths, and they cause significant damage to crops worldwide. These are some images. These are the damages in cotton industry, fruits, and crops. So there are major agricultural losses in fruits, vegetables, and cotton forestry damage from these pests. In Pakistan, we saw last year 34% decline in cotton because of pest issue and environmental issues because of the temperature. So the traditional insecticides have some drawbacks. In Pakistan, traditionally we are using insecticides to spray the cotton fields or different fields, and they have developed the resistance and environmental toxicity over time. But there are some alternatives, sustainable alternatives, which are basically pheromone-based technologies, in which we use the pheromones of insects to control them. And in Pakistan, we need to adopt these new technologies, just like Western countries. So what is pheromone and how it disrupts the mating process of insects? So in insects, usually female, which produce a species-specific pheromone, and this pheromone usually attracts the male for mating purpose. So in the field, we use the synthetic pheromone to confuse the male. So the male cannot reach the female for mating purpose. So pheromones are basically species-specific chemical signals which are released by insects, especially females, to attract male. And mating disruption involves saturating the crop area with synthetic pheromones. These synthetic pheromones usually confuse the males and prevent them from locating the female. And this results in the reduction of reproduction and the population of insect in the crop over time. So lures usually attract, lures are some specific tools which are used in the fields to attract and trap the insects, just like this. Here it has a pheromone, and the males are attracted towards this trap, and then they cannot come out of this. Then there are deed traps which are also used in the fields. There they have the sticky surface and synthetic pheromone, which can attract the male insects, and then they get trapped on the sticky surface. So the core principle is flood the target area with synthetic female sex pheromone, and the male is attracted to dispensers instead of females. And then male antenna also becomes overly loaded and desensitized, unable to detect real female pheromone. And real female pheromone is masked by high background concentration of synthetic pheromone. This is how this technology is used to control the insects in the field these days in Western world instead of using the insecticidal sprays. So these technologies, pheromone-based methods are environmental friendly, they are non-toxic and non-target species. And they are safe to humans, wildlife, and pollinators. There's no chemical residue on the produce and no risk of pest resistance development. And these pheromones have high specificity. They target only pest species. Similarly, major decrease in broad spectrum insecticidal application is the overall result of using these pheromones in the fields. But there are some limitations and challenges of using this technology. First challenge is high cost of pheromone. They are expensive to use. Therefore, in Pakistan, farmers mostly use insecticides instead of pheromones. Secondly, they require separate formulation for each insect, for each pest in the field. So in the world, there are major pheromone producing companies. Like Netherlands, we have Pherobank. This is a major pheromone producing company. Similarly, in Spain, we have Econex. They are producing pheromones on large scale. And at the same time, in UK, Russell, IPM, they are major producer of pheromones. In USA, there is another company, Teresa. This is also a USA-based company. And Sotera is a French company for producing pheromones and supplying. And Nova Agrica is a Greek company. Similarly, Shinitsu is a Japanese company. And what about Pakistan? We have nothing. We are not familiar with this technology. And we have nobody to produce it because you need advanced sciences and advanced skills to make this type of molecules. So 2 years ago, Dr. Shahid Mansoor came to me and proposed that we should make pink bulb pheromone synthesis. We should synthesize the pheromone of pink bulbworm because it's a major problem in the fields of cotton. So this is pink bulbworm. And it destroys the cotton ball like this. So he brought me a sample of this PV rope, which contains the pheromone. It is used like this in the field, and it produces the pheromone. It emits the synthetic pheromone to confuse the insects. So this product was provided by Dr. Shahid to us, and Dr. Sana in my lab analyzed it by doing chromatographic and spectroscopic analysis, and we found that it is basically a mixture of these two isomers. This is the overall synthesis we have done in our lab. And initially we made few grams, and then we are able to make it at 100-gram scale. And now we are working on to produce it on kilo scale. Keep in mind, only small quantity of this pheromone is needed in milligram quantities. And these pheromones are highly potent. So you don't need the large quantities for field. Only 50 kg is sufficient for 50,000 acres of cotton fields. So we did the field trials. Dr. Usman, we provided the pheromone which was synthesized in our lab to Dr. Usman, and then he did their field trials in NIAB, and he reported that our pheromone has same effect just like the imported one. Similarly, Dr. Shahid also checked of synthesized pheromone here in iccbs. In iccbs, we have some plants here. Dr. Shahid is growing his plants here, and here we also get pink bulb occasionally. And these are, we don't have a big field, we have few plants, so we were able to catch some pink bulb with the help of our synthetic pheromone, which was produced in iccbs. The field trials are going on in Multan on a large scale by Professor Dr. Shafqat Saeed, and then we are planning to send our synthetic pheromone to NIPG also in the mid-August to conduct large-scale trials of these pink bulb pheromone. So the next, Dr. Shahid brought me a UK product to control whiteflies. This is another problem in the cotton fields of Pakistan, and Dr. Sana has done the chromatographic and spectroscopic analysis of this, and we are able to figure out the components of this product, and we are in a process of synthesizing this product. And then once we synthesize this, we will request Dr. Shahid to validate the product from our lab with the help of field trials. So the overall conclusion: these loads which contain pheromones are vital for monitoring and mass tracking. Mass disruption is highly specific, environmentally friendly and effective tactic to reduce the mating of insects in the farms, in the field. Together, they form a powerful core strategy for sustainable management of damaging the pest. While challenging exist for this technology, like cost and area, the benefit are reduced pesticide resistance management and safety, because they are safe. They are not toxic like pesticides. Therefore, they are an essential tool of integrated pest management. Apart from the synthesis of pheromone, I want to show Dr. Jamil our synthetic capabilities. iccbs has the ability to synthesize the complex molecules like you have shown to us. For example, this is type of synthesis which we have recently achieved. Dr. Sana and Mr. Talha Fridi, these are two of my students who have recently accomplished this type of synthesis. And this work will be published in the European Journal of Organic Chemistry. Rahila Hanam, my MPhil student, now she's doing PhD in University of Waterloo in Canada. And then phytol-based molecules were also synthesized in our lab by Dr. Sana, and we recently received their vaccine adjuvant results from Advanced Medical Research Institute of Canada. These are the results of their evaluation as vaccine adjuvants from Advanced Medical Institute of Canada. So we can see that our vaccine adjuvant is quite capable of producing cytokines, especially the pro-inflammatory cytokines, so they can activate the immune system. This is another synthesis we have done in iccbs, and these two students were involved in synthesizing this type of molecules. And this synthesis was done by Ms. Norina Naseem. Now she's doing PhD in University of Massachusetts in the USA. And this is another synthesis which is recently accomplished by Dr. Sana, and we are planning to send it for vaccine adjuvant studies in Advanced Medical Research Institute of Canada. This is another work which involves little bit organometallic chemistry, and we have recently submitted it to your Journal of Medicine Chemistry, and it's under peer review since 1 month. This is another multi-step synthesis which was done by Mr. Kamran. He's our MPhil student, and he has recently completed his MPhil, and during his 3-year tenure, he was able to do all this type of synthesize all this type of molecules. My other MPhil student, Mr. Faizan, he has recently made MDP by an alternative method by using different synthetic strategies. And this is the work of Mr. Anas. This is ongoing, and he hasn't completed this work yet. He is in the last stages of synthesis. And the last but not least, this is also that type of coupling which you have shown me the molecules, and you requested me if we can synthesize those, and you can see all these couplings we are easily doing here in iccbs, like isotopic coupling and Hess reaction and hydrogenation reactions and different types of coupling reactions. So this was the overall summary of the synthesis capacity which we have in iccbs. So iccbs is quite capable of synthesizing the molecules which you are proposing, and we are ready to take that challenge. Thank you.
Thank you, Dr. Farooq, for such an amazing lecture. Now I would like to request Professor Dr. Raza Shah, Director in the National Center for Chemical and Biological Sciences, for vote of thanks.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. Brother Professor Salim, thank you so much for visiting us, and I'm extremely grateful to you for your visit and spending time with us, and also discussing research with our colleagues. Before just thanking all the who just make this event possible, let me just briefly tell you about this great institution. This institution is a unique institution in the sense that it is just started in 1966 from a single room, and now we are having 17 research buildings. And the main driving force in this institution and the progress in this institution is the role of leadership. We were blessed with great leadership in the past, right from starting from Professor Srimuzama's Viti. He was a great chemist, a great human being. Then Professor Ataur Rahman took over the torch and led this institution with great vision. And after him, our teacher, Professor Iqbal, he took over the torch and served this institution for more than 20 years. These are the people who are instrumental just bringing institution from a single room to 17 research buildings. And after him, then I took over the torch. And there is so much pressure to just maintain that pace with which they worked, on me especially. So this institution is the only institution in Pakistan which have won 33 civil awards from the President of Islamic Republic of Pakistan. There is no other institution which have received so much decoration. Similarly, this institution is UNESCO Center of Excellence, and we are very much closely working with ComStech on various programs. This is the only institution in the Muslim world which were declared as best research institute twice by the Islamic Development Bank. And probably this is also the only research institute in the Muslim Ummah who have trained more than 100 German students. and they received training in a cutting edge area of research. This institution is just projecting the soft image of Pakistan in the world of science and technology. We have also served this beautiful country in many ways. We have given very top quality graduates to various universities and industries who are serving the nation. In addition to this, whenever there is a need, we just provide our services during the pandemic. We were able to conduct testing to test the COVID-19 virus in the people because, and we were able to establish that facility just in a 2 weeks time because there were no testing facility at that time in the country. And we were asked by the government, now there is a need and you have to deliver. We converted our virology center to a COVID-19 testing facility, and we were conducting the largest number of tests in per 24 hours in the country. So that was actually, and we established that laboratory in just 2 weeks' time. So that shows the capability that this is. Similarly, we were requested by the government that, which kind of strain is now in Pakistan? And we were just analyzing the COVID-19 strain in Pakistan and were just guiding the or policy maker that okay, now this strain is prevalent in the country. Similarly, we have a forensic laboratory. There was no good quality forensic laboratory in this part of Pakistan. So the local government have requested us that you should establish one in the country. And Alhamdulillah, in a very short period of time, a very good laboratory is established that is ISO 17025 accredited. And we also trained the judiciary and the law enforcement agencies. And with that laboratory, the conviction rate, which was before the establishment of this lab, were 4%, and now it is 15%. That is all due to the analysis and the forensic test which we are providing to our law enforcement agencies and the judiciary. And this was appreciated not only at the national level, but also at the international level. Several international media highlighted this, and that was only possible due to the visionary leadership and the great vision which they had. So this was just a glimpses of the research because I just realized that it was not told in the beginning, but this is actually and the success of this institution is the ecosystem of the research. You know that Dr. Farooq just mentioned, the work similarly Dr. Shahid Mansoor is. All this great work is possible due to the ecosystem which have been provided to us, and that is our success. There is a complementary work that every section is complementing another section. All these research buildings, they're built in purpose. They are complementing each other, and they are just working towards to achieve a goal. And that is the vision, because you know that many of the universities have received large number of fundings than us, but they have not tailored it to that level. So it's not the we have received a meager amount of the money, but we invested and every penny is invested at the right place with a vision and with a clear direction. So, and that is only possible with the great mind and great leadership. So with this, let me thank all the people who just make this even possible, especially Dr. Shahid Mansoor, who was instrumental just bringing you here. He was just pushing me very hard to make this trip a success. Dr. Farooq also presented a nice work, our respected teacher, Professor Iqbal Chaudhary, He just reached last night. He was supposed to reach on 8 of this one, but somehow he just reached and he's sitting with us. His support, leadership, and guidance is a blessing from Allah subhanahu wa ta'ala. You know, and thank you everyone for attending this event. May Allah bless you all by this council.
Thank you, sir. Now I would like to request all the participants to gather outside the building for the group photos.